Hello class, my name is Latoya Anthony and I am going to talk about the cell cycle and the regulators of the cell cycle. This is a diagram or a picture of the cell cycle. It tells you the different stages of it, starting from the GO stage all the way to cytokinesis. Um, Before I can talk about what regulates the cell cycle, I want to first talk about the cell cycle. So, what is it? Um, the cell cycle is a set of stages that are ordered. Um, so, they are orderly set of stages, and it starts from the first division to the time of the resulting daughter cells divide. So, there's two major stages. You're looking at interphase and the mitotic stage. Sometimes the mitotic stage includes cytokinesis. Sometimes people put it separate. Um, there are internal and external signals or molecules that controls the cell cycle. This also is where the cell replicates and makes two new cells. So it starts as a parent cell, and by the end of the cycle, there is two identical daughter cells. So the stages of the cell cycle, we start with the interphase stage. This is the beginning of the cell cycle. In the interphase is where the cell spends most of its time. Um, that is because this is where the cell do most of the growing and organelles double. So it spends a lot of time in the interphase here. Um, let's see. It consists of the G1, S, and the G2 phases. So, like I said, it spends most of the time in the interphase stage. Um, in the mitotic stage, this is where mitosis takes place, and mitosis includes the prophase, prometaphase, metaphase, and anaphase. This is also in the mitotic stages where the nucleus will divide. The mitotic spindles do distribute the daughter chromosomes to the two daughter nuclei. Like I said, this is done in the mitosis stage. Cytokinesis, like I said, you can put it with the mitotic stage or you can put it separate, but it is separate from mitosis. Cytokinesis is at the end of mitosis. So right at the end of mitosis is when cytokinesis is present. It's the division of the cytoplasm. The cell membrane is then pinched in the middle, forming the cleavage furrow. This also forms the splitting of the parent cell that forms the two daughter cells that are identical. When we talk about checkpoints is the stage where the cell is examined by the internal and the external signals. This is where it's decided whether the cell can move forward in the division or not. So checkpoints check to make sure the cell is not damaged or there's nothing wrong with it. Nothing wrong. Moves on to the next stage and the cell cycle. There are many checkpoints, but there are three major checkpoints. That's the G1, the G2, and the spindle. The G1 is located at the G1 and the S transition. G2 located at the G2 and the M transition, and the spindle is the metaphase to anaphase transition. So those are our major 3 checkpoints. Um, Again, those checkpoints do allow us time to check for damages and for repairs if repairs need to be done. Signals, the external and the internal signals that we talk about. Again, they can be internal or they can be external. They are regulators in the cell cycle, so we're starting to talk about the regulators of the cell cycle. They control the cell cycle. They are molecules. They can either work as inhibitors or stimulators. They do create metabolic events. Some of the internal signals are proteins like cyclins that can increase or decrease the cell cycle as it continues. And those are going to be done at each of those checkpoints. Again, allowing time to make sure there are no damages in the DNA or the cell cycle. Internal signals. Internal signals is going to be the family of the proteins, and those proteins are called cyclins. These signaling proteins, they regulate the cell cycle, so regulator. The amount present can increase or decrease through the continuation of the cell cycle. So if needed, it's increasing, or when not needed, they do decrease. So the amount does change. It's needed at all of the checkpoints. So each checkpoint is going to have one of these signals that's going to tell the cell cycle to continue, or it tells the cell cycle that the DNA is damaged and it sends it back to the interphase stage, where it's the starting stage of the cell cycle. So if that cell is damaged or the DNA is damaged, sends it back to the cell cycle for repair. Stops it from continuing. The cyclins, their jobs are proteins that bind and activate cyclin dependent kinesis. So call them CDKs. Of course, they regulate the cell cycle. They also initiate processes of mitosis. So it takes the cycle from interphase, takes it into the mitotic stage to mitosis. They are present throughout the cell cycle and can be present in high levels when needed. Cyclin dependent kinases, better known as CDKs, they are very important in regulating the cell cycle. When they are just them alone, they are inactive when they're alone. They are enzymes that are multifunctional. They modify proteins involved in the progression of the cell cycle. They are activated, so they're inactive alone, but when they are bound with the cyclin, they are activated. And then they are assigned to a specific target protein. So them alone, inactive. Binding to a cyclin, make them active, and then they're assigned to a specific protein. And so it has a specific place it needs to go. Okay, so how they are active in the cell cycle. So we're talking about the regulation and the interphase stage. So at the G1 checkpoint, and this is the starting point of the cell cycle, the protein cyclin D will interact with the CDK4 and 6. And what's happening is they are synthesized and initiated in G1 phase. And what they do is they regulate the progression of the cell to the G1S phase. So uh, they check, make sure everything's okay. And then if everything is okay, they will give the okay for that cell to go on to the next phase. It can also react to outside signals like growth factors, nitrogen. So if there are any signals inside, internal or external signals that comes in to this CDKD cycle, then it will stop the progression of the cell cycle, and then the cell stays in the interphase stage for correction. Now, still in the interphase stage, like I said, this is the stage where most of the cell spends its time here. So a lot of time is spent in interphase. When the protein cycling E and A activates the CDK2, so it regulates the start and the growth of the replication of the DNA. Then the cycling E will regulate the G1 checkpoint by triggering the S phase. So the cycling E activates to the CDK2, and then it triggers the start of the S phase. The cycling A will activate with the CDK2, and what they will do is prevent DNA from replicating more than once. So DNA is only supposed to replicate once in that cell cycle. More than one replication of the DNA can cause trouble in the cell cycle. So we want to make sure that DNA is only replicating once. They initiate this synthesis and regulates the S phase completion. So it regulates the completion of the S phase. If the interaction between the cycling E and the CDK2 is not present, the cell is unable to proceed and is stuck in the restriction point. So it's stuck at the starting point in the G1 phase. So this cycling E and the CDK2 interacting between each other to be present is very important for that cell to proceed on to the next phase. So once it gets to the next phase, it's going to go on to the mitosis, so the mitotic stage. Here you're going to find the cyclins A and B, and they're going to interact with CDK1, and they're going to be at the checkpoints G2 and M. Alright, and so these cyclins are synthesized in the S phase. So remember, I talked about that in the last slide. They synthesize. So they're going to be synthesized in the S phase. The cyclin A is going to be activated to the CDK1, and it's going to initiate the synthesis of the cyclin B. Alright, so without the cyclin A, there can't be a cyclin B. And then once they synthesize the cyclin B is going to activate to the CDK1, and it's going to regulate the on-site of mitosis. So it's going to start mitosis, starts that phase. The CDK1B will initiate the cell to enter the mitotic stage. So A will synthesize B, and then B will activate to the CDK1 and then regulate the cell to move on to the next stage, which would be the mitotic stage. Regulation of apoptosis. What apoptosis is, it's the death of a cell that is programmed. Right? So it's regulated by a family of cysteine proteases called capases. The signaling protein responsible for this is P53. And what it's responsible for is stopping the cell cycle whenever the DNA is damaged. All right. So we don't want damaged DNA to replicate. That's how cancer cells start spreading, and that's where you see a lot of cancers from. So when you see So when there is damaged DNA starting to replicate, it's going to stop that cell, and it will try to send it back to interphase stage. So it tries to send it back for repair. If coming through the steps again, if it's not able to be repaired, then apoptosis is initiated, which is, like I said, the programmed cell death. The importance of regulators. So regulators are important in the cell cycle because they make sure there is proper homeostasis of the cell. So make sure that cell is balanced. It's crucial to the survival of the cell. So it's also responsible for detecting DNA damage, and it repairs it. Also, it's responsible for preventing uncontrolled cell division. Like I said, as in cancer cells, prevent those cancer cells from dividing and multiplying and becoming too many cancer cells, which can be harmful to the body. All right, so again, that is the cell cycle and the regulators of the cell cycle. There are many more regulators. I kind of just went over some of the most important ones, but there are many. And this is just references. Didn't mention is when things can go wrong with the regulators. So there is such as dysregulation cell cycle, and what that is a place where majority of tumor cells will live. So majority of regulating the cell cycle is to prevent cancer cells. So majority of dysregulation are associated with cancer cells because the regulation stops the cells from the cancer cells from duplicating and replicating. So if there is a dysregulation of those cancer cells, the normal cells that are damaged and the DNA will not go into the cell cycle arrest, and then it won't go back to the interphase stage and prevent the or repair, and then it don't start the apoptosis whenever there is something wrong with the DNA. And one of the cycling that I did find is the cycling in the CDKs that, of course, like I said, the cycling will activate with the CDK, and that is what the regulation regulates the cell cycle. But there is the CDK2 that will bind with it's the CDKN2. So remember I talked about the P53 protein signal that will signal the cell to go into apoptosis when damage can't be repaired. So there is a protein 16 and they bind to the CDK4 and the CDK6. And what that protein 14 and 16 does is it protects the protein 53, the P53. It prevents it from being broken down. So it allows that P53 to stay active in the cell cycle. So whenever that P14 and that P16 is not present, the P53 cell protein is broken down, so it doesn't work as it should work. And what happens then is you got those cancer cells that's damaged. They don't go back for repair. They don't go into apoptosis. They continue to replicate, and then it's mostly associated with like the head and neck cell cancer, lung cancer, and bladder cancer. So the breaking down of the P53 is associated with those cancers. Can be related to genetics. Um, so that is why the regulators of the cell cycle is very, very important.